So looking at the quiz itself, I'll go through any questions specifically, unless Amanda, you had any questions about anything as I go. Feel free to stop me and then we can talk about it further. So to begin, just kind of a review of in prokaryotes, transcription occurs in the blank, and then transcription occurs or translation occurs in the blank. So you'll see that there's a distinction in the locations of transcription versus translation. Um, and then there's a true or false question for question two, which is a certain gene or DNA sequence always results in the same final protein product. Hopefully, you found that in the T.A. Wansheng's video that genes or DNA sequences might not always result in the same protein. That's because of several mechanisms that he talks about. Then looking at question three, we go back to our favorite DNA codon table. Um, so you're looking at GAT um, in the middle of a gene, and the sequence mutates to GCT. So using the codon table, you'll see if that changes um, anything about the translation of the RNA molecule. So then, in question four, how can the same RNA transcript produce different amino acid sequences? Same or similarly to question two, you'll probably be looking at the same parts of the PowerPoint, looking at how RNA molecules that come from the same gene can encode for different amino acid sequences.
Me go back to number three.
Yeah, for sure.
So when it says DNA, it's gonna go to um, mRNA, right?
Correct. Yeah.
So the GAT would become C um, UA, right?
Right. Right.
So that would be LEU. I don't
Okay.
know what it is, but
Yeah. Yeah.
uh,
Yeah. Leucine. Yeah. Perfect.
okay, leucine. And then if it mutates to GCT, that would be CGA,
Yes. CGA.
uh,
Yeah. So if we
which
look
is
there, sorry.
arginine, right?
Yes, exactly.
Um, in the answer questions, I believe that I put B, but now I don't think that's right.
So what I'm realizing might also be the okay, I see. So this question should be a bit more specific because that's how I would have gone about it. But now that I'm rereading it, it says the DNA codon GAT. That's possibly the sequence for the coding strand instead of it being the template strand. So if it were the coding strand runs 5 prime to 3 prime as well. So it's going to be the same as the mRNA, but here it would just be what GAU on the mRNA. Does that make sense?
Okay, okay,
So GAU, that's where we get our the ASP there, or asparagine.
okay,
So I think your original answer was correct, actually.
okay. And how would
Yes.
we de determine that though? Like if there was a question like this on the exam, how would we know whether to go off the coding strand or the template strand?
Gotcha. So it should say in the question. Um,
Okay,
but worst comes to worst, for this question specifically, you see that how can the same RNA or sorry, looking down here, translation would stop prematurely. Obviously, that wouldn't be the answer just because it doesn't turn into a stop codon. Like I would just do
right.
process of elimination here where and then here you have there would be no effect on translation. I mean, there's an amino acid substitution. And
Hmm.
then a shift in an open reading frame, that would just result from an addition or subtraction of nucleotides in the DNA sequence.
Okay,
So yeah, you would just have to do a process of elimination. But
okay,
yeah, I would like to think that on the exam, it would specify.
perfect.
Yeah. Really good question, though. Yeah. I didn't even notice that. Cool. Okay. So that's for question three. Um, yeah, once again, looking at four, you'll look at the different mechanisms for RNA transcripts to produce different amino acid sequences. Then questions five and six, I believe it's both five and six, um, utilize some bioinformatics tools that are pretty popular these days still. You're looking at BLAST, P, and UniProt. So BLAST is a great tool for actually inserting a DNA or protein sequence and then finding out what the protein is. So you'll find the answer for question five, and then question six builds off of question five, and that you're going to use the protein name and insert that into UniProt, which is a great tool for instead of actually finding what sequence encodes for the protein, it does a great job in synthesizing information on the protein, like the result protein. So these tools kind of build off of one another. So I think Wansheng also included some slides on where you'll be looking, specifically on the UniProt page for the behavior or like the characteristics functions of the protein.
Yeah, the,
So
the guide was really helpful.
okay, good. Yeah, that's good because there's so many like windows on those websites, I feel like, or so many different
Yeah, and
sections.
then a lot of the lists are pretty long. So
Oh, definitely. Yeah.
following the guide actually got me to the right answer, so that was good.
Okay. I'm glad that that was helpful because doing this the first like I'd say even like 10 or 20 times, you're still trying to figure out what what you're looking for. So I'm glad that helped. And then question seven, in eukaryotes, which component directly recognizes and binds to the TATA box of a gene's promoter? You should be able to get that from the PowerPoint. Then in question eight, okay, so you're given the mRNA sequence 5 prime to 3 prime. Which amino acid will the complementary tRNA carry? So you're still going to be looking at the codon table, not the tRNA sequence, because the codon table goes from mRNA to amino acid. Um, so this is just talking about what amino acid the tRNA will actually carry. That wording is a little confusing, but since the given RNA sequence is 5 prime to 3 prime, you should just be able to use that on the codon table. So then on question nine, a mutation changes the mRNA codon from GAA to GAG. What is the most likely effect on the protein? So once again, you'll be using the codon table with these like exact sequences, GAA and GAG. See if that affects the actual amino acid that the tRNA will carry. And then that could lead to one of these four mutations that are explained on the video. So then question 10, which molecular complex catalyzes intron removal in eukaryotic pre-mRNA? So this is a process that occurs in the nucleus before the mature RNA reaches the cytoplasm for translation. So you should get that answer as well from the PowerPoint. Then for question 11, which feature is unique in eukaryotic gene expression? You'll notice that three of the answers are unique more for prokaryotic gene expression, but one has to do with RNA maturation because that only occurs in eukaryotes because there's a distinction between like the cytoplasm, which makes up most of the cell, and then the nucleus where all the cool stuff takes place. It's kind of like a warehouse almost in my opinion. And then finally, question 12, a microRNA binds to the 3' UTR of a target mRNA. What is the expected outcome? I think that's also talked about on the PowerPoint. Um, but this is my favorite. This is more what I study. So, Amanda, do you have any other questions about the quiz?
Nope, I think I got all my questions answered.
Okay, awesome. I'm glad that helped. What about in general? Anything else?
I was working on activity 7, but um,
Gotcha.
I'll join Dr. Holcheck's hours tomorrow so she can help us with that.
Gotcha. Okay, cool.
Appreciate you explaining the questions.
Oh, of course. No, I'm glad I could help. That is, yeah, that was definitely a little confusing. But yeah, thanks for joining.
Thank you.
Yeah,
Short meeting.
I know, right? Yeah. Sometimes it's like this with like if not many people show up.
Well, I hope you have a great rest of your week.
Okay, thank you. You too, Amanda.
All right. Bye.
Bye. So we're going a little out of order, but I do want to talk about some things on the PowerPoint that TA Wansheng prepared. So just looking at the learning goals for this recitation, it's to understand central dogma as a whole, how DNA goes from RNA or to RNA through transcription and how RNA gets translated into a protein. Um, there's also some details on the prokaryotic transcription steps. And the third goal is to explain ribosomal translation processes. And then finally, to compare expression control in prokaryotes and eukaryotes. So looking at the central dogma, like I mentioned, DNA is transcribed to mRNA, and then mRNA is translated into a protein. Now, something that's important that we will look at in this recitation is that a single gene can yield multiple proteins. And that's due to several processes, but one of the main of which being alternative splicing. So I would like to go down here and just start talking about transcription initiation. So when looking at the um, prokaryotic side over here on the initiation, you have the sigma factor that's accompanied with RNA polymerase just upstream. And when I say upstream, that's more 5 prime or to the left in this orientation of the actual gene that's going to be transcribed. On the flip side, or also looking at transcription initiation, you have these regions called Tata boxes. And this is an important area for transcription factors to bind and also recruit RNA polymerase to the actual transcription start site. I also want to mention that the transcription start site is different than the translation start site. So, or like the start codon that initiates translation. As you might remember, messenger RNAs, mature messenger RNAs, are composed of both exons and introns, with the first exon having the translation start site or the start codon of AUG, which encodes for methionine. But that starts translation. However, upstream of it, there's also a 5 prime untranslated region, usually in eukaryotes. So what you're going to see is that you're going to have a non-coding region upstream of the actual start site. Then continuing to go, we talk about elongation and termination. And what we see is that the mRNA strand is complementary to the template strand, meaning that the mRNA strand also has the same sequence as the coding strand. Then Wansheng talks a little bit about these row independent and dependent hairpins and helicases. So then looking at the eukaryotic promoter region or the TATA box also, we have TATA binding proteins or TBPs, and they're within the transcription 2 factor or transcription factor 2D, and that's what binds to the actual TATA box. And so the DNA will actually bend um, due to the TATA binding protein, and then it positions the transcription factors and up from upstream enhancer regions to all interact so that it recruits RNA polymerase to the gene so that transcription can occur. And the sigma factor is bacterial, um, and activators bind to distant enhancers. So overall, there's a bunch of proteins that all play a role in increasing the levels of transcription. So then looking at the translation overview, we see that ribosomes read codons 5 prime to 3 prime. Start codon is methionine, so it sets the frame of the protein as well. As you might guess, all of the genes or exons are all like the number of nucleotides are all divisible by 3. And that's so that when the ribosome actually translates the RNA, we have the exact number of nucleotides that's necessary for a specific number of amino acids. So then tRNAs are these cool RNA molecules that create this three-dimensional structure that interact with specific amino acids depending on their sequence. And the tRNA sequence is going to be complementary to the mRNA sequence. Like I mentioned when we were going over the quiz, the mRNA sequence is what you're always going to be using when looking at codon tables. You never want to use the complementary tRNA anticodon sequence. There might be such thing as anti-codon tables, but usually us researchers just use codon tables, meaning that we look at mRNA to protein. So stop codons are UAA, UAG, and UGA. So looking at translation, we have initiation. Methionine is the start of the polypeptide chain. So we have a methionine initiator tRNA that will interact There's also a ribosome around here, as you can see. Um, and within the large ribosomal subunit, the tRNA will um, bind to the mRNA, and then we have the start of the translation of this specific gene. So during elongation, you can see that three tRNAs are kind of interacting with the ribosome at the same time at positions E, P, and A. So it first interacts with, or it first goes into this like A region, then it goes to P, where it actually the amino acid that's brought by the tRNA is added to the polypeptide chain, and as you can see, this tRNA that doesn't have an amino acid anymore will actually leave the larger ribosomal unit because its job is done, and then it can be recycled. So elongation will continue until it reaches like a stop codon. Then termination will occur so that the resulting like polypeptide chain can leave the ribosome and then undergo some folding mechanisms. So, of course, looking at prokaryotes versus eukaryotes, since eukaryotic cells have the distinction between the cytoplasm and the intranuclear space, we see an actual separation of transcription and translation. And this is important because eukaryotic cells are usually part of larger organisms. Um, well, not usually, there's a lot of single-celled eukaryotic organisms, but um, the the benefit of having a separation of transcription and translation is that you can have more fine-tuned regulation of genes. As you might remember from prokaryotic gene regulation, looking at operons, it's usually always on or usually always off. There's a couple like in between stages where you have some transcription, a medium amount of transcription, and a lot. But when you separate um, transcription and translation, I like to think of it as they're just being the volume dial having more settings. So it goes from like 3 to 10 or 3 to even 100. So there's a lot more proteins at like involved, leading to a larger genome, which is sometimes a negative for especially single-celled organisms and their survival, but the actual level of fine-tuned uh, genetic regulation uh, usually benefits eukaryotes. So here's a helpful table comparing prokaryotic versus eukaryotic gene expression mechanisms. And then finally, I did want to mention all of the mutations that um, directly happen to DNA sequences, and that might lead to different genes being expressed, or to the gene being expressed, but different protein products. So, silent mutations are ones that have no effect on the protein sequence. So, as you can see here, it looks like this GTA, the A in the third position is mutated to a T, but the looking at the codon table, you'll see that both of those codons encode for valine. So, that's a silent mutation because there's no actual phenotypic change. Now, missense mutations have amino acid substitutions. So the in this CCC codon that encodes for proline, we have a mutation in the first position that changes it to an A, and now you have threonine. So we actually have a phenotypic change, meaning that the protein coming off of the gene changes a bit. And even one amino acid substitution can have great effects on the actual protein because, as you might remember from secondary, tertiary, and even quaternary structures, the folding of a protein really matters for its actual function. So, one change in an amino acid can lead to great differences in the resulting protein. So nonsense mutation, as you can see here, this tyrosine has a mutation on the final um, nucleotide, making it TAG, which will then be UAG on the mRNA, which encodes for a stop codon. And that will just lead to like a truncated protein, is the term that researchers use. And that is just a nonsense protein, meaning it has no function at all. And then finally, frameshift mutations are probably the most detrimental. So you have the insertion or deletion of nucleotides that changes the entire protein sequence usually. So you can see here that if you have, um, I think it's a subtraction of these two amino acids, this sequence goes from SVPY to SVLL. So you see that multiple amino acids are changed. Then looking at whole chromosomal mutations. You have duplication of genes, deletion of whole, genes or loci, and then you can have translocation, which can be super scary when looking at um, genetic regulation because certain chromosomes have specific genes, and that results in uh, usually like normal levels of regulation for a given gene. But when it moves to a different chromosome, it's going to have a completely different um, set of regulatory mechanisms. For example, um, the one I can think of is that if you have a gene that promotes cell growth that's in a specific location on a chromosome, so that's only turned on when cells need to grow and divide, but it gets translocated to another chromosome that has housekeeping genes, so these are always on, you're always going to have cell growth and division, and that can be lead to oncogene behavior or promoting tumor development. So overall, genetic mutations are usually not good, but of course, we evolved from genetic mutation. But that, of course, took millions and billions of years in order to get here. So then just looking at the importance of gene expression, kind of like I just talked about. Now, alternative splicing is also another way for eukaryotic genomes or transcriptomes to increase greatly. So when we talk about genome, we talk about all of the genes. When we talk about transcriptomes, the idea that you might have is that we're talking about all of the transcripts or all the possible RNA molecules. Now, obviously, RNA is the kind of like middleman between genotype and phenotype. So when you have a lot more possibilities of RNA, even from the same gene, or when you have several possibilities of different RNA molecules from the same gene, you'll find that you can have variant phenotypes, which can increase the level of diversity and pretty much strengthens the overall survival of a given population. So here we see alternative splicing where all five exons are included, but then in this middle transcript, exon 3 is spliced out. And then in this one on the right, exon 4 is going to be spliced out. So that will lead to three different protein variants that might have different, um, slightly different jobs within a cell. So this is kind of a cool way for eukaryotic cells to get the most out of their genome. Then microRNAs are non-coding short RNAs that inhibit translation of mRNA by binding to somewhere within this 3' untranslated region. And what's interesting about this microRNA is that it has proteins that come along with it that will actually splice RNA molecules. So these microRNAs, since they're, 22 nucleotides, they're somewhat specific, kind of like primers are. But these proteins that splice up RNA use microRNA as a tune or as a way for fine-tune genetic regulation, specifically post-transcription regulation, because we already have a mature messenger RNA here. So transcription is over. So that's why we call it post-transcription genetic regulation. And I find this stuff super interesting because this is specifically what I research in the model organism, Cynorhabditis elegans, which is actually where microRNAs were discovered. So if you have any more interest about that, feel free to reach out to me too. I have some great review articles that you could read. But then we just have guides for using the bioinformatic tools BLAST and UniProt. So best of luck on your recitation quiz, and feel free to reach out to me if you have any other questions.